In this video project, we will investigate the protein alpha-synuclein and its association with Parkinson's disease. Nearly 1 million Americans are affected by Parkinson's disease, which is characterized by the accumulation of Lewy bodies in the substantia nigra. Lewy bodies are abnormal buildups of alpha-synuclein found within nerve cells that contribute to disease. Parkinson's is a degenerative disorder of the central nervous system that disrupt basic motor functions, resulting in tremors among other symptoms. We will highlight the structure of alpha-synuclein, discuss the mechanisms of aggregation of alpha-synuclein that contribute to Parkinson's, and conclude with current therapeutics. Alpha-synuclein is a 14 kilodalton protein that consists of 140 amino acid residues, of which residues 1 to 60 represent the amino terminus, 61 to 95 the so-called non-amyloid beta component, and residues 96 to 140 the carboxy terminus. The amino terminus is rich with amino acids lysine and threonine. Particularly, it contains apolipoprotein lipid binding motifs conferred by a KT-KEGV sequence, which is predicted to form amphiphilic helices that confer the propensity to form alpha helical structures during membrane binding. The NAC contains a highly hydrophobic motif that comprises amino acid residues that tend to self-associate and lead to the development of the aggregate, which confers beta sheet potential. Finally, the carboxy terminus of alpha-synuclein contains no significant secondary structure and is highly acidic and negatively charged. The C terminus may be responsible for preventing aggregation of Lewy bodies, as evidenced by the operation of the carboxy terminus of alpha-synuclein and alpha-synuclein 112, 114, and 97, which involve disruptions in the carboxy terminus, and these removals or disruptions of the carboxy terminus seem to promote aggregation. Interestingly, alpha-synuclein seems to exist in chemical equilibrium between three distinct structures: beta-like structures, mechanically weak folds, and random coils. Of the three conformations, beta-like structures are directly related to alpha-synuclein aggregation, suggesting that a critical concentration of alpha-synuclein in beta-like structures is needed to trigger oligomer, fibril, and Lewy body formation. There are many proposed mechanisms that may explain the abnormal aggregations of the alpha-synuclein protein found in Lewy bodies, but we will highlight one proposed theory. A lab from Penn has proposed that the abnormal aggregation of alpha-synuclein is formed from a crucial sequence of 12 amino acid residues, 71 to 82 in the middle of a hydrophobic component of the protein. The similar beta-synuclein protein in humans is almost identical to alpha-synuclein, but lack these 12 crucial amino acids and fail to aggregate, as indicated by the green label. To further confirm the importance of this hydrophobic region in fibrillogenesis, a single residue in the middle of this region, alanine 76, was substituted with either a positively arginine or negatively glutamic acid charged residue, denoted as A76R and A76E mutations. Adding a charged amino acid in the middle of this hydrophobic residue sequence in vitro greatly decreases the rate of alpha-synuclein aggregation, indicated in red. Lastly, synthetically made proteins comprised of this 12 amino acid stretch self-aggregate to form filaments as indicated by OD400, which indicates degree of fibrillization. These 12 residues self-associate and even in the presence of alpha-synuclein will promote fibrillization. There have been many therapeutic proposals, but a majority tend to focus on disrupting the oligomeric process. For example, there are several mutations in the KTK repeat region of the alpha-synuclein gene that may contribute to increased oligomer formation, which represent possible avenues for therapy. However, we will focus on therapies involving the phosphorylation of alpha-synuclein on serine 129, which changes its hydrophobicity and charge distribution, leading to increased aggregation. Thus, regulation of this phosphorylation process provides a therapeutic option. This is confirmed in a blot representing alpha-synuclein aggregation. Chloramphenicol acetyltransferase is used as a negative control for alpha-synuclein, and beta-tubulin is a loading control. The S129A mutation impedes phosphorylation of serine, leading to lower levels of aggregation, as is evidenced by a decrease in the signal of phosphorylated alpha-synuclein for S129A. Whether through proposed therapies involving mutations and others listed here, the variable properties of alpha-synuclein protein make for diverse pathways by which Parkinson's may be tackled.